In this video, we're going to see if it's possible to identify these tiny microscopic single-cell organisms. It's not as easy as you might think.
Micromysteries.
I encountered my latest micromystery in an unlikely place. While walking home from the dentist, I noticed that a drainage ditch beside the road was filled with duckweed, covered end to end with green. I love sampling duckweed, as I often find plenty of organisms living on the leaves and roots. I always have my trusty plastic container with me, so I grabbed some duckweed and water and headed home to the microscope. The first thing I saw was this population spike of ciliates, what we call a bloom. I've seen blooms before, they're beautiful. And while the term bloom normally refers to algae, it is increasingly used to refer to any rapid population spike, including ciliates and flagellates. But something about these organisms made me hesitate in attempting an ID. They looked familiar, but something was off. The more I watched, the more questions I had. Identifying these cells involved multiple imaging techniques and the help of experts more than 5,000 kilometers away. Who doesn't love a mystery? The sheer number of organisms in a microbial bloom can be mind-boggling. I think the number and diversity of blooms I encounter in Thailand can be chalked up to our climate, hot and wet. These are just a few of the blooms I've come across. I think you'll agree, they're amazing. But let's get back to this bloom from the roadside ditch. After marveling at its sheer beauty, my natural instinct was to figure out what I was looking at. Sometimes I recognize something immediately, but much of the time, I need to look for clues. I consider anatomy and morphology, the shape and structure of the organism. Then physiology and biochemistry, how does it move, eat, reproduce? And finally, ecology and behavior. Where does it live? What role does it play in its world? Of course, there are genetic and molecular indicators too, but for the most part, those are out of reach for a microscope hobbyist like me. I usually aim to identify organisms at the genus level. That's often enough to understand how they live, feed, and reproduce. Getting to an exact species usually requires tools and expertise well beyond my reach. Even genus is sometimes a stretch. There were two different organisms in this bloom. The larger, longer ones are Paramecia, a very common single-cell organism that most people are probably familiar with. It's these smaller ones that I found interesting. So where do I start? In this case, my first clue was the way they looked. They were bell-shaped and had cilia at the open feeding end of the bell. Cilia are tiny hair-like structures that help organisms swim through water or sweep food particles inside. Under the right lighting, you can see the patterns these cilia make, and that can tell you a lot. When I see something bell-shaped with cilia lining the bell, it immediately leads me to a specific group of organisms, Peritrichs. Peritrichs are really cool, and I see all sorts of them in almost every sample. They're found pretty much everywhere. I might see one, or I might see 1,000. They can be individuals or colonies. They can attach to surfaces with a contractile stalk, or a stiff, unbending one. Stalks can be singular or branched. The variety is almost endless, but they often have that bell-shaped front end, or something relatively similar, lined with cilia to help it feed. And that's what I was looking at in this mystery bloom. But Peritrichs are almost always sessile, meaning they don't swim around. They usually attach to surfaces with a stalk or adhesive disc, spending their lives content with their view, able to use their cilia to create a vortex bringing food inside. They reproduce through binary fission. Sometimes they become a meal for something larger. So when I see Peritrichs, I mostly see some variation of this. But clearly the ciliates in my sample were not attached to anything, not a single one of them. Could they really be peritrichs? The real question is, can peritrichs swim? And the answer is yes. In fact, they can be very good swimmers. But why would an organism that is normally attached to a surface swim? There are three primary reasons why a peritrich would swim. First, they can just become detached. Sometimes while being collected or placed on a slide, they can accidentally separate from their stalk. Or maybe a microscopic worm or other microscopic life form knocks them off. In this case, I might see one or more peritrichs swimming around amongst mostly sessile groups. Second, some peritrichs undergo a stage in their lives when they purposely detach themselves and swim, a stage known as the teletroch. This helps them spread out and colonize new areas when conditions change. And third, a very small set of peritrich genera spend their entire lives as free swimmers, never attaching to surfaces. And well, this is where I made my first mistake. I misunderstood the word teletroch. I thought it meant any free swimming stage of a normally sessile peritrich, and so I included both those who accidentally and purposely detached in my definition of teletroch. I posted a short video of this bloom to an online microscopy group and described it as blooms of paramecia, teletroch peritrichs, and other ciliates living in duckweed. So why is this important? When IDing an organism, it's not only important to get the name right, but also to describe it correctly. Without a correct description, there is little chance of correct ID. The first response I got was from a well-known Australian expert in the field, David Seymour. He suggested that these were not teletrochs, but instead colonial peritrichs that had become destalked. I replied that it would surprise me, as I had looked all through this sample and there hadn't been a single stalked peritrich, only these swimmers. Could every one of them have been destalked? At this stage, I had only posted low magnification dark field views because, well, they showed off the beauty of the bloom. For those counting, that was my second mistake. Trying to ID microorganisms without high magnification detailed views can be difficult, even impossible. So I told David that at high magnification, I was able to see cilia at both ends of these peritrichs, not just around the front end. With that new piece of information, a side conversation in the comments opened the possibility of these being teletroch peritrichs, and David was able to point to posterior rings of cilia on these peritrichs that might support this. When peritrichs enter the teletroch stage, they absorb their stalk and then develop a posterior ring of cilia used for swimming. So our search for an ID might have been left right there, were it not for another sharp-eyed viewer who noticed that some of the peritrichs in my video were undergoing binary fission. They were reproducing. He suggested that if this were true, these wouldn't be teletroch peritrichs, as teletrochs do not divide while in this stage. He wondered if this could be a population of free-swimming individual peritrichs instead. Based on this discussion, David shared his excellent drawings of two groups of free-swimming peritrichs and suggested I consider that the bloom was one of these organisms. It was time to take a higher magnification video to try to match my organisms with David's drawing. These organisms spun and moved quickly at high magnification, which made filming detail difficult. So I added some methyl cellulose, essentially a very low concentration of wallpaper glue, to slow them down without harming them. When David saw this video, he believed that my bloom was the peritrich Opisthonecta, which has a distinctive tuft of cilia called an opisthobal membrane. As long as I had them under the microscope, I decided to film them using a lighting technique called fluorescence. This involves staining the organisms with the dye acridine orange that binds to DNA and RNA, and then shows up under fluorescent light. And there it was, the U-shaped nucleus that was so prominent in David's drawing. That clinched it. I was excited to not only have found something new to me, but also to have it correctly ID'd, the result of awesome teamwork from a vibrant online community in different parts of the world. Sure, I made some mistakes along the way, but the drive to understand what I was looking at and the knowledge shared by others allows me now to confidently state that you are looking at a bloom of the Peritrichs Opisthonecta. I hope you find them as beautiful as I do. Thanks for joining us on today's look inside the Microcosmos. We hope you'll consider subscribing to our channel so you won't miss our next adventure.